All right, let's get right into it. The decade between 2010 and 2020, wow. This was when synthetic biology basically went from its awkward, ambitious teenage years to a full-on revolution. It was this make-or-break decade, you know? A field that was absolutely swimming in hype had to finally prove it could actually deliver real, world-changing results. So, how to go? Let's find out. At the start of the 2010s, the buzz was just inescapable. Synthetic biology was promising this incredible future where we could literally program life. Think about it, designing cells to be tiny factories, or living sensors, or even data storage devices. The potential was just massive. But, as this quote from some top researchers back then points out, the field was kind of known more for its hype than its actual results. So the big question on everyone's mind was, could the science actually deliver on these huge promises? And yeah, that's really the central question we're digging into here. Could this field, which was just packed with brilliant ideas, get over the enormous technical hurdles needed to engineer biology in a way that was actually reliable? Or was it all just going to be a super interesting but ultimately impractical academic exercise? Well, the decade definitely started with a bang. There was this one achievement in 2010 that really fueled the dream and gave everyone a sneak peek of what was truly possible. This was a huge moment. I mean, a team at the J. Craig Venter Institute basically booted up a bacterial cell using a genome they built completely from scratch in a lab. It was proof that we could write the entire genetic instruction manual for an organism. We're talking millions of letters long. The ability to write the code of life was suddenly becoming a reality. But as you're about to see, writing the code is one thing, getting it to run properly, that's a whole different ballgame. And yeah, that was the big problem. Despite all that early excitement, the field started smacking right into some very hard truths. It turns out the messy, complex, and totally unpredictable nature of life is incredibly difficult to tame and engineer. See, the dream was always to engineer biology just like we engineer electronics, with predictable, standardized, reliable parts. But biology, it was the complete opposite. You could have a genetic part that worked perfectly in a test tube, but then you pop it inside a Wiming cell and nothing, it would completely fail. It was just so context-dependent, messy, and frankly, incredibly unreliable. Here's a perfect example of what I'm talking about. Researchers would spend ages designing a genetic circuit. It's kind of like a biological if this then that switch, and they'd get it working perfectly. But then they'd take that exact same code and move it into a slightly different strain of bacteria, and poof, it would just break. Without that standardization, without that reliability, the whole dream of engineering biology was basically stalled. So how on earth did the field get past this massive roadblock? Well, the answer wasn't some single eureka moment. Instead, it was this rapid-fire development of a totally revolutionary new toolbox. And these tools made engineering biology faster, cheaper, and way more predictable than anyone thought possible. The path forward really came down to accelerating this engineering cycle. You design something, you build it, you test it, and then you learn from it. Now, at first, every single one of those steps was painfully slow and manual. But then this wave of new technologies came along and just supercharged the whole process, letting scientists test thousands of different ideas all at once. And look, if we have to pick just one technology that changed everything, it's got to be this one. CRISPR wasn't just another tool in the box. It was the breakthrough of the decade, not just for synthetic biology, but for all of the biosciences. It handed researchers this incredible new power to edit the code of life with unbelievable precision. Now, most people know CRISPR as a kind of genetic scissors for cutting DNA, right? But synthetic biologists, they got really creative with it really fast. They adapted it for so much more. They basically turned it into a programmable dimmer switch, which let them turn genes up or turn them way down. This gave them precise control over a cell's entire internal machinery. It was a game changer. But CRISPR wasn't working alone, not by a long shot. At the same time, the cost of just making custom DNA totally plummeted. It literally became cheaper to just order a gene online than to try and make it yourself in the lab. New automated methods made snapping all those DNA pieces together like they were Lego bricks. And all this was backed up by powerful computers that let scientists design and analyze thousands of genetic blueprints all at the same time. So, armed with this incredible new toolbox, the field went from, staring down those hard truths to suddenly delivering an absolute explosion of achievements. The hype was finally becoming reality. And the pace of discovery was just staggering. Seriously. What started as just a little trickle of milestones in the early 2010s had become a total flood of breakthroughs by the end of the decade. And it was touching pretty much every area of biology. Let's just pull out a few key moments to show you what I mean. In 2013, scientists completely rewrote the genetic code of an organism. That's a huge foundational proof of concept. Fast forward to 2016, and we had software that could automatically design complex biological circuits for us. And then by 2019, these tools were being used to create smart cancer-fighting cells, you've probably heard of them, CAR T cells, that could be programmed to hunt down tumors more safely and effectively than ever before. And the applications that came out of this were just mind-blowing. Scientists engineered bacteria that could live inside your gut and actually record what was happening in there. They figured out how to use DNA as a data storage medium, way denser than any hard drive we have. They created these cheap little paper-based sensors that could quickly detect viruses like Zika. And they programmed yeast to produce really complex molecules like opioids and cannabinoids, basically turning tiny microbes into living pharmacies. It's wild. Now, all of this incredible science is one thing, but the ultimate proof, the real validation that all that hype was justified, is what happened next. The research didn't just stay in the lab. Oh no, it exploded into a massive new industry. And the result of that? A multi-billion-dollar industry, creating everything from new medicines and materials to sustainable foods and fuels, and it was all built on the backs of these newly engineered cells. This was the ultimate payoff for a decade of really hard, foundational research. And what's so cool about this story is who built it. This wasn't just a bunch of big old companies buying into some new tech. This industry was actually founded and grown by the very same PhD students and postdocs who were doing all that painstaking research at the lab bench. They literally turned their science into world-changing companies. It's an amazing story. Which, of course, leaves us with one last, really fascinating question. The 2010s. That was the decade we finally built the toolbox to reliably engineer life. So now that we have the tools, the real question is, what incredible things are we going to build with them in the decades to come?